selenium 200 years ago using very, very primitive technology. He didn't have a mass spec or ICP-MS or anything, atomic absorption spectroscopy or spectrometer or anything at those days, but he did, do some very, very nice, elegant work to show and confirm a new element. All right, next slide, please. So where do we get selenium? We get selenium from our foods, particularly foods like broccoli, shown here in green florets, and then Brazil nuts, garlic actually has quite a bit of selenium. And selenium smells almost like sulfur. It is virtually, just by smell, you cannot distinguish sulfur and selenium. They're very similar. We don't need to require a lot of selenium. That's why it's called a trace element. As you can see here, selenium is, requirements change with age, and with physiological state in women, we need a little bit more selenium. And it also has selenium has an upper limit of about 400 micrograms per day. Although that's a very conservative upper limit, we can go up to about 1,000 micrograms, that is 1 milligram per day, and not see any toxicity. But we can go up to 400 very safely. But what is really important in the next slide, now this selenide gets converted by an enzyme called selenophosphate synthase to form what is called selenophosphate. Now that selenophosphate is then charged on to a tRNA that is supposed to be the second tRNA for serine, or has a serine attached to it, okay, charged on to this tRNA. Now, when the selenophosphate is charged on to this tRNA, that tRNA then becomes the selenocystenyl tRNA. And the gene that encodes this particular tRNA for the second tRNA for serine, that is the specific one that is used for this incorporation, is encoded by a gene called TRSP. Okay, I highlight this because we have now developed models in the lab where we have made a genetic knockout of a mouse strain. So this mouse does not express this gene, so that we can actually completely ablate all the selenoproteins. Now, what happens is when you get this tRNA, unfortunately you cannot see what is behind this clearly. What happens is that tRNA recognizes a codon, which is a stop codon. So all in your biochemistry textbooks or biology textbook, you have been told that UGA is a nonsense or a stop codon. That is actually only half true. The UGA can also function as a selenocysteine codon only when under special circumstances in the body. And that depends on a specific hairpin loop structure in the 3' end of the messenger RNA. It's called this, there is a, unfortunately, I can't even show you my pointer, but there is a green circle there behind the red stop sign, and that is called BP2. And that BP2 binds to this secondary structure in the mRNA, and that structure is called selenocysteine insertion sequence. And this is present in about 25 or so of the mRNA in our genomes. In humans, it is 25. In mice, it is 24. So about 24 genes actually have this particular element in their 3' end of the RNA. And so what happens is when that 3' end structure is present, the UGA codon becomes a selenocysteine codon. And so when in all those selenoprotein mRNAs, you will have multiple UGA codons. So the penultimate UGA is always the UGA that is used for selenocysteine incorporation, and the last UGA is where it stops. So the system can recognize between a UGA that is selenocysteine UGA versus a UGA which is the authentic stop codon. So this is where biology gets very interesting and molecular biology gets very interesting. Okay, so that's how selenocysteine is incorporated. And once it is incorporated, it acts decay process, reduces all the selenoproteins. And as a result, what happens is when you reduce all the selenoproteins, that is when most of the effects are seen in our body. Okay, if you go to the next slide, I'll show you what these selenoproteins do. So selenoproteins actually help in regulating the oxidative tone of our cells because all our cells in the body are constantly being activated by a variety of different ligands, and those ligands then increase what is called oxidative stress. So we have very nice systems in our cells that reduces oxidative stress and brings them back to homeostasis. That is called redox mechanisms. So glutathione is a tripeptide that is indirectly related to selenoproteins and is also a co-substrate for selenoproteins. So selenoproteins help in glutathione metabolism. There is also, many of our proteins, enzymes like phosphatases, kinases, all the transcription factors have thiols or cysteine thiols in their active sites or DNA binding sites. And all those DNA binding sites have to be kept in a certain redox state, and selenoproteins help in doing that. Then there is also what is called protein folding. Proteins that are translated need to be folded really well, otherwise the misfolded proteins can actually cause disease. For example, in Alzheimer's disease, that's what happens. So this misfolding can also lead to stress in cells. So selenoproteins help in folding them because some of these selenoproteins are disulfide oxidoreductases, which have that enzymatic activity and help in this. We have shown recently, I'll show you a slide in the next few minutes, where selenium helps in epigenetic regulation. So the question that my lab has been asking for many years now is, do these selenoproteins play a role in immunity and disease pathogenesis? What happens in those? So next slide, please. We know that selenoproteins have a role in immunity. Many of our work from other laboratories throughout the world have shown that selenium deficiency adversely affects the ability of immune cells to put up an immune response. So for normal immunity, you need a very good selenoprotein pool. So as I told you earlier, we have certain cells and change their phenotype such that they can now become really helpful in an immune response. And this is seen in case of, say, viral infections or bacterial infections and so on. We and others have shown that selenium supplementation helps, and some studies in humans given about 100 micrograms of selenium actually helps increase immunity to certain viral infections. And the same is true with COVID as well. So in HIV, we have also shown that supplementing patients with HIV, with selenium, can actually decrease the viral load and improve the immune parameters, and that occurs through regulation of viral infection. So in the next slide, please. One of the things that we have done is, we've focused on a particular cell type called macrophages. I'm sure many of you have heard about these cells. These are cells that are present in virtually every tissue in your body. Okay, you take the lungs, we have alveolar macrophages. You take the liver, we have what are called Kupffer cells. In the central nervous system, we have what are called microglial cells. And in spleen, we have splenic macrophages of different kinds. And then in the bone marrow, we have a lot of macrophages. And these macrophages actually help in a variety of different physiological functions, also in pathological functions. Now, they have beneficial functions, but they can also do harm if they are activated and the activated macrophages stay that way without getting resolved. So this is exactly where inflammation and resolution comes into picture, because these macrophages can differentiate into different types of macrophages. For example, a naive macrophage, shown here as M0 macrophage, can become a highly pro-inflammatory macrophage. Okay, we often call it a rowdy macrophage. These rowdy cells now are converted because of a lot of inflammation, say bacterial infection or viral infection. And that cytokine response you see, which actually in COVID that you see, is because of these M1 macrophages and multiple different cell types too. However, you can actually tame these rowdy macrophages and make them really very nice and cool macrophages. Okay, so these macrophages then are called M2 macrophages. Now, that conversion of M1 to M2 can happen in your body through certain cytokines and also have certain nutrients. And this is what my lab showed. One of our seminal findings in the field was that we showed that you can actually take a pro-inflammatory macrophage and make it into an anti-inflammatory macrophage in the body by adding selenium to the system. Now you can ask me, so what? Okay, so you convert a rowdy to something that is not a rowdy macrophage. Now what happens? If you look at the next slide, what we did was we showed that during the onset of inflammation, okay, it can be an infection or a vaccine injection, for example, you get inflammation. That inflammation is actually very beneficial. So oftentimes we call this particular inflammation, particularly during vaccines or any infection. So what we have shown is that this onset of inflammation, bringing about resolution, is a very critical event in your body. And if you can make that event quickly, you can actually have a very nice adaptive immunity set in your body, and that will be protected. So for this to happen, you need a lot of nutrients as well. So eating healthy actually helps this process. Okay, if you go to the next slide, I'll show you what we do in the lab. Currently, we use this we study the inflammation and resolution in colitis, in helminth, parasitic infection, bacterial infection models. We also do anemia models, and then we look at cancer. So next slide, please. Before I do that, I want to give you a quick overview of metabolism, particularly fatty acid metabolism. There are two different types of the polyunsaturated fatty acids. One is called the N6 fatty acids. The other one is called N3 fatty acids. The N6 fatty acids are, for example, linoleic acid, arachidonic acid, and so on. Whereas the N3 fatty acids are also called omega-3 fatty acids. And these fatty acids get metabolized in your body when you take, dietary fat, for example, particularly linoleic acid, which is an essential fatty acid that we cannot produce. When you take that through, eating foods that have, say, for example, sunflower oil or safflower oil, and all those, you get those fatty acids, and those become converted to arachidonic acid. Now, this arachidonic acid can go through a variety of different pathways called the cyclooxygenase pathway, lipoxygenase pathway, cytochrome P450 pathway, and produce a lot of very interesting metabolites in your body, okay, in our bodies. Now, the same thing happens when you take N3 or omega-3 fatty acids. These fatty acids, particularly EPA and DHA, called the icosapentaenoic acid and docosahexaenoic acid, are produced particularly in, say, some plants or in fish. And these are fish oils and considered to be very beneficial in the body, okay, for cardiovascular or in decreasing your triglycerides or cholesterol and so on. And again, they all go through metabolism in the body, and all those metabolites that are produced from your diet dietary fatty acids or fatty acids that you take actually help in your general health. So what we have shown is selenium can impact some of these pathways. You don't have to remember these pathways, but just the fact that selenium, through certain mechanisms I'll show you in a bit, can impact these pathways. So if you go to the next slide, what you see is, particularly with arachidonic acid, what happens is arachidonic acid through the cyclooxygenase 1 and 2 enzymes, which are the targets for your non-steroidal anti-inflammatory drugs like ibuprofen or paracetamol or naproxen and all these drugs, indomethacin, all these drugs actually inhibit cyclooxygenase 1. So cyclooxygenase 1 acts on arachidonic acid, produces a prostaglandin called prostaglandin H2. Now that prostaglandin H2 can become a variety of different prostaglandins depending on which tissue it is produced in. In most tissues, in most cells like say macrophages, for example, we can see a lot of prostaglandins like prostaglandin E2, thromboxane A2, prostaglandin F2, and so on. What we did was many years ago, again, I'm not going into the details, giving you a very fair overview, 35,000 feet overview of what we have done over the years, multiple papers. What we have shown is that when you take a macrophage, treat it with selenium, and inflame those cells that have selenium, now skew this pathway of arachidonic acid more towards this green prostaglandins, the prostaglandin D2 and prostaglandin J2, rather than the inflammatory prostaglandin. And this mechanism happens because of many transcriptional circuits that are regulated by selenium. As a result, what happens is instead of producing inflammatory prostaglandins, you start making anti-inflammatory prostaglandins. And so we have termed this mechanism as icosanoid class switching. And this icosanoid class switching is very, very, very beneficial to reduce inflammation and begin resolution. If you go to the next slide, please, just to give you a brief overview of what these prostaglandins that are very good for you are. These are prostaglandins called prostaglandin J2 and 15-deoxyprostaglandin J2. These are highly electrophilic prostaglandins, and this is where some chemistry is important because these molecules are extremely interesting molecules. I have spent almost half of my adult life in trying to chase these prostaglandins and understand their mechanisms, and we still continue to do this. And these prostaglandins can impact a variety of different pathways, different transcription factors, epigenetic mechanisms of histone methylation, because as you know, histone methylation or histone acetylation, deacetylation are all extremely important mechanisms in your body. And so what happens is when you add these prostaglandins or you produce those prostaglandins, the effect can be felt even at the level of the gene expression mechanisms. And what we showed here many years ago, this was done by one of my former postdoctoral fellows, Ravi, who showed that if you take a histone acetyltransferase called P300 HAT or histone acetyltransferase and treat it with prostaglandin J2, you can actually inhibit the enzyme. As a result, what happens is genes that are to be expressed because of the histone acetyltransferase activity will now be suppressed. And those genes particularly are inflammatory genes, and we shut off the gene itself. So it is like flipping a switch. And when you turn off the switch, no genes are expressed, particularly inflammatory genes are not expressed, and that reduces inflammation but increases resolution through other mechanisms. So we continue to work on this, particularly in a cancer context in the lab. And what we do is in the next slide, you can see we use a lot of, as I told you, even though I'm a biochemist by training, I love seeing doing a lot of chemistry as well, organic chemistry, bioorganic chemistry. And one of the instruments that we use that I have in the lab is a triple quadrupole mass spectrometer, which will actually give you identities of many of these organic molecules by not just a single, you know, uh, um, uh, um, fission event or a single fragmentation, but also multiple fragments that are produced, and you can actually quantitate. In one shot, in one injection, you can quantitate about 100 metabolites in these, and the sensitivity of this instrument is in the picogram level, and so it's actually extremely sensitive instrument. And what we do is this is a LC-MS system where the analytes are passed through. It can be blood samples that are extracted through a C18 column, or just by Folch extraction method, just solvent extraction, and then these are taken in the mobile phase. And the mobile phase, in this case, we use about 80% methanol water system. And there is a HPLC column here, which takes and separates the analytes and sends it to the mass spec. And the samples are all put in an auto sampler, so you don't have to keep injecting. They're all done. If you just program everything and keep it there, it is kept at, it's a refrigerated auto sampler. And so it is injected, and you get the identities of all the compounds when you come next day. So you have the data ready when you come the next day morning and see what are the compounds in your system, in your samples. So the mass spectrometer uses what is called multiple reaction monitoring tools, so that we can actually, quantitate each of the analytes based on the fragmentation from the molecular ion to the daughter ion, such that even if an analyte, migrates at the same retention time in an HPLC, it could be a mixture, and that mixture can be resolved using the mass spec very nicely. So that's what we do in the lab constantly, all the time. So using this mass spectrometer, what we have done is we have tried to understand how does selenium actually help in inflammation and resolution. So we go to the next slide. We did a huge metabolomic study where we took, the urine samples of the mice that were inflamed, and we saw that when you treat them, this is a principal components analysis. It's a statistical method to cluster the samples based on how they behave and what kind of patterns you get in terms of similarities and dissimilarities. And using the statistical method, what we show here is on the left side, you can see that as you increase the levels of selenium in the diet, you start seeing the clusters change. Okay, the green ones or the red ones, the green ones, the triangle ones, and the up triangle blue ones are all different, and they cluster differently, suggesting that they start producing different metabolites. So to understand what these metabolites are and how they are different, we decided to go back to our macrophage model, where we were able to treat them with LPS, which is a bacterial product, which increases inflammation, like you would have during a bacterial infection. And we saw that when you culture the macrophages with or without selenium and treat it with LPS for various different time points, like from 0, 2, 4, 6, 8, 20 hours, you start seeing changes in metabolism. And what we saw was that without selenium, you actually make a lot of glycolytic products, and you reduce the production of ATP through down-regulating the TCA cycle. As a result, what happens is there is more glycolysis, there is more stress in the system because they have to crank up the glycolytic pathway to meet the energy demands. But when you add selenium, the broken TCA cycle, or Krebs cycle, for example, the broken TCA cycle is repaired, and then you start seeing the reduced cranking up or reduced glycolysis, but increased TCA cycle, which now produces more ATP. And the ATP is important because it gives you the energy to for multiple physiological mechanisms that you need energy for. And that helps in reconstituting the system. So this was recently published, and if anyone wants to read this paper, I'll be happy to send it to Dr. Devika, and she can circulate it. So next slide, please. So overall, what we have done is we have shown that if you take the M1, the rowdy macrophage, and treat it with selenium with all these cyclopentanone prostaglandins, the prostaglandin J2, for example, you can actually make them into the M2 macrophages, and that induces what is called wound healing response and attenuates inflammation. And this polarization of macrophages is a very important thing in reducing inflammation and increasing resolution. Again, I have not gone into details, but I've given you a fair overview. But I'll switch gears now a little bit and tell you another story with the omega-3 fatty acids. So if you go to the next slide, please. So as you know, omega-3 fatty acids are, in many foods that we take, like flaxseed, for example, walnuts, kidney beans, chia seeds, and so on. And in milk or in baby foods, you name it. It's become almost a fad to include omega-3 fortification in multiple, foods that we eat. And in fact, if you go back to Mahatma Gandhi's book, he actually tells you in early 1900s, when he was touring India, he found that in villages wherever he toured, where people were taking flaxseed, the disease levels in those villages was actually low compared to those places where there was less flaxseed consumption. The diseases were higher. He makes this comment very nicely in his book. That is perhaps because one of the reasons, in part, could be the levels of omega-3 there. So you can see we have been exposed to this for a very long time, but our food habits have changed over the years, and that's probably one of the reasons. But anyway, it is all coming back now. It's a circle, right? Life circle, it keeps going on. So anyway, going forward, the question is, why take fish oil? Why take some of these foods? If you go to the next slide, please. You can see that fish oil, or in capsules, or eating 2 servings of fish per week, you get particularly 2 fatty acids, EPA and DHA. And now these fatty acids actually boost immunity. They reduce cholesterol, and they also have anti-inflammatory and anti-cancer effects. Multiple people have shown this. But again, we don't understand the mechanisms clearly. So if you go to the next slide, please. The mechanism that we know is that these omega-3 fatty acids actually reduce metastasis, and what they do is they can actually reduce the ability of some of these cancer cells to not proliferate. As a result, what happens is it reduces the proliferation of cancer cells, and the cancer slowly regresses. But it may take a long time, provided you keep continuing to take omega-3. So if you go to the next slide, please. For example, if you look at a variety of different fish, for those of you who don't eat fish, you take fish oil capsules. Now, there are multiple different fish that have different amounts of omega-3. So not all fish are the same. So just by eating a fish, if you think you'll be fine, that is a myth. I wouldn't go with that. You need to know how much of omega-3 you are consuming. So best is to probably take one of these fish oils that are very pure, and then that might help. So next slide, please. So one of the projects that we did very early on was to take a macrophage cell in culture and treat it with EPA, like we do with our body, take EPA capsules. And then give EPA to human volunteers, take the serum from them, and do mass spectrometric studies to see what kind of compounds are produced. And what we found was there are very interesting compounds that are produced. And this is seen not just in macrophage models in the Petri plate, but also in humans taking EPA supplements or fish oil supplements. And if you go to the next slide, what we found was that when you take fish oil supplements, the EPA is converted to these interesting compounds, the icosanoid prostaglandin J compounds that I told you earlier. But these are slightly different because it has an extra unsaturation because of the starting fatty acid is different, EPA, which has 5 double bonds. So it makes a prostaglandin called prostaglandin J3, and this prostaglandin J3 can isomerize and dehydrate and all that, but you get those final prostaglandins, and these prostaglandins are called the cyclopentanone prostaglandin J series. Now, the question we asked was, do these have any health benefits, particularly in cancer or in leukemia? I'll go through very quickly some slides in the next few minutes. So next slide, please. What we decided to do was to study leukemia. As you know, leukemia, the reason why we did leukemia was leukemia has much better markers compared to other cancers. And we have a big group here that studies leukemia. So it was mostly from a convenience point of view also. We were getting a lot of samples and such. So that's why we selected leukemia. And then in leukemia, what happens is there is a normal hematopoietic stem cell. This is a normal cell which gives rise to all your different cells in the body, okay, like platelets, your other white cells, and monocytes, macrophages, basophils, everything. Okay, but however, what happens is when these cells are undergoing constant cell division, slowly there are some mutations that are introduced in your body, in these cells, these hematopoietic stem cells. So the normal stem cells, now when they acquire some mutations, I'll tell you one mutation, they now become what is called a leukemia stem cell. Now, those leukemia stem cells can undergo proliferation and give rise to what are called leukemic blasts. And those leukemic blasts are the ones which actually are the ones which cause the disease. So you have, when you look at a cancer patient, a leukemia blood cancer patient, they have very high levels of white blood cells, okay? And that is called leukemogenesis. Now, if you go to the next slide, please. These leukemia stem cells are a key target for new types of therapeutics, because if you look at the leukemia therapies, they are pretty old. What we use currently are about 20 to 30-year-old therapies. So we really need new therapies, and we need to know more about these leukemia stem cells. These leukemia stem cells are also very intelligent, okay? So they have evolved to, if you give them a drug, they have evolved with mechanisms to efflux the drug so that the drug is immediately thrown out of the cancer cell, stem cell. Or they acquire certain mutations such that the drugs become inactive, just like a antimicrobial resistance in bacteria. And then they also have epigenetic mechanisms such that they can shut off genes or increase genes and so on. So these cancer cells are also very intelligent. So we decided to take on this challenge to see if some of our fatty acid, omega-3 fish oil fatty acids, derived compounds, endogenous lipid mediators like prostaglandin J, can impact these cancer stem cells or leukemia stem cells. The next slide, I'll tell you what we did. So the central hypothesis was that cyclopentanone prostaglandin, particularly delta-12 prostaglandin J3, shown here, the structure for chemists, you will like this structure. There are two electrophilic centers here and here. Unfortunately, I can't point my pointer, but you can see where the electrophilics are, C9 and C11. And so this is a prostaglandin J3. It is also called 9-deoxy delta-12, 9-12 prostaglandin D3. Has cis and trans unsaturation and so on, which makes it very interesting to study as well. And this one, the hypothesis was this particular compound will ablate the leukemia stem cells by activating a certain pathway called P53. So we go to the next slide. We use two models. One is a retrovirus called friend erythroleukemia virus. And what it does is it causes infection. And when it causes infection, you can see the spleen becomes huge, shown on the left side. We also did a second model where we expressed a human oncoprotein in those normal stem cells in the body of the mice. And we show that it leads to leukemia where the spleens again become big. The spleens becoming big is called splenomegaly. And that is a commonly seen sign in leukemias where you can actually touch the side under your right rib, very close to the liver, and you can actually palpate it very nicely. And when you palpate it, you know there is inflammation, just like you see in jaundice patients. So we go to the next slide, please. What we did was we injected the mice with the friend virus. And after we did for a week, we let the leukemia develop. And then we started giving them for 1 week. After that, we started the injection with delta 12 prostaglandin J3 at a very low dose, okay, 0.01 to 0.5 milligram per kilogram body weight. And remember, these mice are about 30 grams roughly in weight. And so we gave them this compound, and we also used a standard, drug called cyclopamine, which is used to be used, but now not used because it has teratogenic effects and all that. And what we saw was just like what you see there, the vehicle control had huge spleens, but cyclopamine actually brought the spleen size back to normal. You go to the next slide. In 7 days, okay, so we also looked at a technique called flow cytometry we use in the lab, where I'm sure some of you have heard this technique. It is a technique that uses fluorescence-based analysis of a variety of different immune cells. And what we do is, unfortunately, you can't see the flow cytometer on the left side. That is the flow cytometer that we use. And what you do is, depending on the cells that you are interested in looking at, you use certain antibodies with fluorescent tags. And those fluorescent tags then will fluoresce when they go through the flow cytometer. And it analyzes cell by cell, one cell at a time. And then what you do is you get a forward scatter and a side scatter, which tells you the size of the cell as well as the granularity of the cell. And so you can actually distinguish different, what is going on there in the sample. This is used commonly in medicine and immunology. If you go, they take your blood and they subject it to flow cytometry analysis. And so if you go to the next slide, using flow cytometry, we analyze the spleens of these mice that were treated with prostaglandin and also infected with the friend virus. And what you see here is that the infected with just the vehicle control, the spleens were huge, while the ones treated with prostaglandin at 0.05 and 0.01, you could see a difference, and 0.05 was very effective, almost same as an uninfected mouse. But if you looked at the white cell counts, which is the key biomarker for leukemia in the peripheral blood, you saw that there was a huge increase in white cell counts, WBC counts, that reduced when prostaglandin J3 was injected. And when we do flow cytometry, we do not see these stem cells in there, suggesting that the stem cells have all been wiped. And if you see histopathologically, if you do sections of the spleen, you can clearly see the spleen structure returns back in the treatment group, suggesting that treatment with this prostaglandin actually brings back homeostasis, kills all the cancer cells, and then restores homeostasis. So that was one model. If you go to the next slide, what you see here is, actually, this is how the mouse looks when you open it. You can see uninfected mouse, the spleen is very small, you can't even see. But look at the spleen in an infected mouse on the left side. You can see big spleens. Now, when you treat it with the compound, delta 12 prostaglandin J3, it returns back to almost like an uninfected mouse. This is the best way to show the effect is there already, right? You can all appreciate this. So if you go to the next slide, we decided to take this model and make it even more human relevant by using a human mutation that is commonly seen in patients of chronic myeloid leukemia. The mutation that is often seen that many of the pathologists look for when anyone comes in with high white cell counts is they do a a smear, and they do karyotyping analysis and so on. And they see that if you have any mutation, and one of the mutations they look for is chromosome 922 translocation. So where chromosome 9 and chromosome 22 break apart and then join at chromosome 22 to form what is called a Philadelphia chromosome. The reason it was called Philadelphia chromosome, as you can see in the next slide, is because this was first noticed by two physicians called Peter Novell and Dave Hungerford at the University of Pennsylvania, which is close to us here in the state of Pennsylvania, in Philadelphia. So that's why it's called Philadelphia chromosome. And Janet Rowley, who was later awarded the National Medal of Science, a very top honor for scientists in the United States by President Obama, showed that this was actually a target, and it brings about what is called dysregulated kinase activity because of that small mutation and that breakage of the chromosome that can be used to treat using tyrosine kinase inhibitors. So this is the karyotyping analysis, and you can clearly see the the chromosome 22 there that has changed, and this can be easily seen by a pathologist very nicely in the lab. So we used this model, and what we did was we took that BCR-ABL that is formed and made a fusion protein. And what we did was we expressed that gene using a virus, as you can see in the next slide. What we did was we took the mice, we took the bone marrow from these mice and infected them with a virus that expresses this BCR-ABL human protein. And we then took those bone marrow cells and injected them into a recipient mouse that was irradiated. Or what we do is we irradiate the bone marrow such that we kill all the bone marrow cells, such that when you do a bone marrow transplantation, this is typically also done in human patients. When you do a bone marrow transplantation, the donor-derived cells now establish very nicely and engraft in the recipients. And slowly, over a period of time, about 2 to 4 weeks, we see the disease. And this disease is very similar to what we see in human patients. So in the next slide, please. We decided to now treat these mice that were transplanted with the cells that had the human mutation. And what we see is when you transplant those with human mutations, you can clearly see that the treatment group actually has very low stem cells that you can see by flow cytometry on the right-hand side here. And those are different markers that we use for stem cells. One is called stem cell antigen. The other one is a receptor called stem cell factor. It is called KIT. And then the other one is because the virus also expresses a green fluorescent protein, these cells can also fluoresce in a flow cytometer. And so these cells are also GFP positive. So we use what is called differential gating. And because of those differential gating, we can actually see and specifically quantitate these cells, leukemic stem cells. Now, when you treat it with prostaglandin J3, those leukemic stem cells actually come down. This was a very nice result for us, a very positive result that told us that this may be effective. So if you go to the next slide, please. I'll just quickly share with you the data where we have when we give these mice the stem cells, the cancer stem cells, those mice do not live beyond 14 days, about 2 weeks from the day of transplant. But if you treat those mice about 1 week after transplant with the leukemia cells, and then treat them every day for 1 week, okay, with prostaglandin J3 at very low doses, you can see these mice survive. And we get almost 100% survival. This tells you that this prostaglandin treatment is probably very effective. So the question we asked was, can we, this was a pure compound being given. So we asked the question in the next slide, can we take omega-3 supplements in the diet and still get protection from omega-3 in the diet? If you go to the next slide, I have a few slides. I may have to jump quickly, But what we did here was, you all probably know that fish oil is given as a therapy for reducing triglycerides, and that is typically given at about 3.4 grams to 4 grams a day. And what we did was, we first made sure that people taking this omega-3 at that high pharmaceutical levels or pharmacologic levels to reduce triglycerides produce prostaglandin J3 in their serum. So we did a mass spec study of human volunteers taking this, and we found that they do produce prostaglandin J3, which was very great. And then what we did was, we can't give 4 grams to a mouse. Okay, so we had to reduce the levels based on the energy levels. And what we did was, we did what is called allometric scaling of the doses based on the surface area of a human and a surface area of a mouse, and brought it down. So if you go to the next slide, we made a diet and fed the diet to these mice containing about 1.8% EPA in the diet, which equals to about 3 grams or 4 grams of omega-3 in a human being. And what we did was, we gave these mice either oleic acid, which is a control, or icosapentaenoic acid EPA, and put these mice on diet for about 8 weeks. And then we activated this pathway by giving them a shot of LPS, just so that the fatty acids that are esterified in the phospholipids will all get released and produce these in this bloodstream, so that we could take just the red blood cells and do what is called an omega-3 index, as well as quantify delta 12 prostaglandin J3 by LCMS in the blood. Please, next slide, please. So when we did this, we found the omega-3 index, as you can see here, increased significantly in the liver, spleen, RBCs, suggesting that the mice were eating very well, of course, and all the omega-3 got incorporated in the membranes very nicely. So if you go to the next slide, we now challenge these mice with leukemia. And what we see is, when you challenge, if you go to the next slide, please, this is just showing that they produce omega-3 derived J3. So what we did was, we took these mice on the diet, transplanted them with the bone marrow transplant with the oncoprotein, and then we did a lot of studies. Just because of time, I want to just go to the last, just keep skipping the slide to about, to this slide, and yes. So what we showed here was, when you give them EPA in the diet, what happens is, the stem cells that are cancer-specific stem cells actually go down. Okay, and when you treat these mice with also a non-steroidal anti-inflammatory drug, it blocks the effects of the fatty acid that we have given in the diet. And then only when we give prostaglandin J3, we rescue that effect. The moral of the story is that if you take supplements, you have to be careful what drugs you take, because the drug and the supplements can interact and can negate the effects of the supplement. So just blindly taking supplements and blindly taking certain anti-inflammatory drugs may not really do good for you. This is exactly what we proved in this paper. Okay, so the question now is, I'll just jump to the last slide here, slide number 51. If you just jump, please. Next. Yes, the question is, you may ask, what happens in humans? So first, what we did was, we took normal human bone marrow and from healthy volunteers and cultured them with this prostaglandin at pretty high levels. And we did not see any effects on their differentiation, which means the toxicity effects are likely to be very low for the prostaglandin if we were to use prostaglandin as a drug therapy. And then what we did was, we got multiple samples from leukemia patients. Can you go to the next slide, please? And what we did was, we took leukemia patient samples from AML and what is called a CML patient samples, and we treated them with prostaglandin J3, and this is all again in a Petri plate. And what we did was, we did a apoptosis assay for cell death, And what we saw was there was a high level of apoptosis when treated with these prostaglandins, suggesting that these patient samples responded very nicely to these drugs, to the prostaglandin as a drug. So very likely we could actually do a clinical trial. So what we have done now is, if you go to the next slide, we have now started a clinical trial to investigate this with our physicians and oncologists here in our university, where a research that started very much with undergraduates and PhD students, postdoctoral fellows, is now gone to the bedside. And we are trying to do this two ways. One is with a pure prostaglandin J3. Other one is to give them very high dose fish oil, like you do to reduce your cholesterol and triglycerides, and see if it affects the patients. So currently we have some patients that are recruited in this model, in this study, and we are trying to do this. If it works well, we would like to do this in a large scale and all that, and so that'll be nice. As a scientist, I think this is a great reward to see your therapies go from bench to bedside. This is one of the best awards I can get, is a effective therapy to save people. I will conclude my seminar with a very simple conclusion slide. That if you go to the next slide, please. Again, this is what we are trying to do with the molecular response in patients, but this is not important. We'll go to the next slide, please. So what I told you was selenium in the form of selenoproteins reduces inflammation through mechanisms involving anti-inflammatory and pro-resolving lipids. Similar to this, fish oil is also metabolized in immune cells to novel anti-inflammatory and anti-cancer compounds such as prostaglandin J series of eicosanoids. And what we showed was LSCs, that is leukemia stem cells, can be targeted by apoptosis by this compound. And it activates multiple intracellular signaling pathways. I didn't want to go into details, but this is a compelling reason to start a clinical trial, and we've already done that. We've started. With that, I would like to conclude showing you a picture of all my lab mates and lab colleagues, my students, who have all, over the years, have been in the lab and helped with all these projects. Many collaborators here at Penn State, a variety of different institutions and so on. These are, and funding from NIH. I will just leave you with one last picture. I think you will all like it. Please, next slide. You can see here your principal was my classmate in this picture. This was taken in 1988 at Yuraja College when I was also a student with your principal, Dr. Devika, and I'm there hiding behind somewhere. And so thank you once again for the invitation to Dr. Devika and to Dr. Subraya and other administrators. Now, this is not important. So thanks once again. Thanks to all of you. I'm really sorry we took a little bit longer, but my apologies. I should have checked this earlier. But anyway, I'll be happy to answer any questions. Thank you.
Thank you, sir, for the interesting presentation. According to World Health Organization, inflammation and cancer are on the top global sources of death. We appreciate that this mysterious area of research was explained so well. It was truly a stimulating presentation, sir. Thanks a bunch for sharing your time and knowledge, and our sincere apologies for the technical issues. And my sincere thanks for all the participants for your patience. Now the session is open for queries. The participants can unmute their mic or use the chat box for the same.
So there are a few questions there. Let me read out. First question is, we see reports that selenium interacts with statins, barbiturate, anticoagulants, niacin. Can it be administered to elderly? What dose level can be beneficial? So yes, that's a very good question. Selenium actually interacts with statins in a beneficial manner. Actually, statin treatment has been shown to reduce selenoproteins through a mechanism that involves the what is called the farnesylation or isoprenylation of tRNA for the selenoproteins. As you know, HMG-CoA reductase inhibition would also reduce isopentenyl pyrophosphate. Isopentenyl pyrophosphate is required to mature certain tRNAs. And when you take statins, what happens is that isopentenyl pyrophosphate goes down, you don't mature tRNA. As a result, you get decrease in selenoproteins and also other proteins too. So that actually causes the muscle tenderness or myalgia that you typically see in statin patients. So it does help to increase selenium levels as well as even in anticoagulants. In vegans in India, there's a question: does garlic can be sufficient? No, garlic is by itself is not very sufficient. Um, so you need to have additional sources of selenium because garlic contains some selenium but has more sulfur. A question on will omega-3 obtained from seaweed? Yes, actually, plant-derived omega-3s can also be effective. Like ALA can be effective in producing EPA-derived because ALA can get converted to EPA, albeit at low levels, but still can be beneficial. Yes. Thanks, Dr. Rana. Selenium on parasites, helminths. Yes. Plasmodium. Yes. There. We are actually interested in the Plasmodium story, although there has been very few publications, and they are not very conclusive as to the role of selenium in Plasmodium or malaria infections. And this is really nice because we can actually do this and show. So that is something I'm interested in doing in the future. Can you suggest good sources of selenium for strict vegetarians? Oh, well, yes. I remember I had a slide there. Flax seeds, chia seeds, walnuts, and broccoli. All those are, and then Brazil nut is the most highest amount of selenium that you can get. One nut is enough to give you about 400 micrograms a day. So that is a great one. Okay. Any other questions? I don't see any other questions. Feel free to ask me any questions. If I know, I will tell you. If I don't know, I will check and I will give you the answers. Any questions? I really don't know in dementia patients. The question is selenium for dementia patients. What doses can be helpful? I don't think there are good studies to show yet selenium might be useful. If, that is something we will have to do more studies. But taking about 100 micrograms of selenium per day, once people turn about anywhere after 50, is probably not a bad idea. Of course, you don't want to do that just blindly because you want to make sure how much selenium you have in your system before you actually supplement. Because you may be already having very high levels of selenium, and on top of that, if you now supplement, it could actually cause, it may not be beneficial. So one has to be careful how much selenium we do. So when we do this in patients, we first look at what is their blood levels of selenium, we have certain biomarkers, and only after checking that, we either put them on selenium or not. We just can't do that blindly because it can lead us to, misinterpretations and wrong interpretations. Any other question?
Sir, there's a question. Where can we reach you for future queries?
Oh, okay. My email is ksp4 at psu.edu. You can email me at this email, and I'll be happy to answer any questions.
Thank you,
Okay, or
sir.
you can connect. You can connect me on ResearchGate as well.
Sure,
But
sir.
email is fastest. Yes. Any other questions?
Sir, good evening, sir. I'm Metro speaking.
Hi,
Yeah.
Petro. Good to
Hello,
hear
sir.
you.
Yeah, it was such a wonderful session, and I congratulated the organizers, Devika Madam and team, for arranging this wonderful talk. And really, it was really inspiring that how your work has progressed, sir. And then we're looking forward uh, that it will be successful, the clinical trials, everything.
I hope so.
Uh,
Yes.
yes, I'm sure it will, since it's quite promising.
Yes.
So, sir, the PGJ series, that the 12 delta PGJ which you have used, did you synthesize it and then are you using it, or
Yeah.
um,
So we have synthesized this molecule. Uh, there is a there's a paper, Angivantic Chemi paper, that we published a few years ago in collaboration with Professor Casey Nicolaus' lab at Rice University in Houston, Texas. It is a 26-step synthesis, and this molecule has been made in a GLP GMP grade. So we are hopeful that we can take this forward. We have some issues with the stability of the molecule and so on, particularly just it summarizes just exposure to the fluorescent light, tube light. It can I summarize. So we are trying to work on some of that aspects of it, but we have successfully achieved full total synthesis, and it is active, biologically active molecule. Only thing is we want to make sure it is stable over a period of time, and all that stability analysis, all this. It takes a long time to get all these things done, and we have to now go in for FDA approvals, and it's not easy to get FDA approvals before we start a clinical trial. So that's why we started the first phase with the EPA, because that is a dietary intervention, so that it is much easier, and EPA has already been FDA approved. So all we had to do was get get a new IND for a different indication, that is investigational new drug application for a different indication, which is cancer instead of hypertriglyceridemia, and then do this study. So that FDA approval was pretty easy, but again, compliance issues, many different things. It's very strict here, so we don't know. That is a challenge, but we'll see.
okay, thank you, sir. So, are you administering through orally, or is it by what way is
Yeah.
the drug administered?
So prostaglandin J3 that I showed you here is all injections through intraperitoneal injections daily in PBS, phosphated buffered saline, or just saline. Whereas EPA is given orally. Yeah.
All right, thank you, sir. So I wish all the success and look forward to hearing
Thank you.
the success stories in all this
Hopefully.
major, definitely in all this scientific magazines. Definitely, it will be
Hopefully.
highlighted.
Yes.
Looking
Yeah.
forward, sir. Thank you.
Thank you.
Thank you.
Thank you. Any other question? Oh, okay. So there is There are two questions. Can selenium be useful in immunomodulation, especially in the context of COVID and post-COVID scenario? Yes, certainly. Yeah, there have been some studies, and we are also doing the studies here. Selenium can be beneficial because you have massive inflammation during COVID. It's called the cytokine response, systemic cytokine response, or inflammatory cytokine response, that you can reduce to some extent with selenium supplementation. Yes. What makes chromosome to mutate in leukemia? I don't think we know, Dr. Devika. It is this constant cell division that makes the chromosome 9 and 22 labile, and then they break off and then form this. Yes.
Okay.
Yeah, we don't know the reason. Yes.
Okay.
Yeah, there are multiple mutations. It's not just the only mutation. There are about, to date, particularly for myeloid leukemias, there are over 50 to 60 some odd mutations. And in our clinic, we test for quite a few of those mutations.
Okay.
And all of them manifest in one form or the other of myeloid leukemia, or even lymphoid leukemias, which also have similar mutations. Yeah.
Okay.
But we don't know what causes the mutations.
Thank you. Sahiti,
Any other?
if there are no more queries, conclude it.
Thank you, sir, for answering the queries. If there are no more queries, we can conclude the session.
Thank you once again,
Yes.
Dr. Devika and Sahiti and others.
Yes, sir.
Great
Honor to
interaction.
have you here, sir, on the talk. Listening
Thank
to
you. Hopefully, when I visit Mysore, I'll visit in person, Shraddha Vilas, and then maybe I'll be able to
Yes,
interact.
sir. We're looking forward. Now I
Continue, Sahiti.
Now I request Rutu Aradhya, first year MSc, to render the vote of thanks.
Good evening, everyone. So we have reached the end of this knowledgeable session. On behalf of Shraddha Educational Institution, Mysuru, I am Ritu C., first year MSc Chemistry student, Shraddha College. I think it's a great honor and privilege to propose the vote of thanks on this memorable international webinar. Let me first of all start by giving a glory to Almighty God for making today's occasion a resounding success. I owe special gratitude to today's speaker, Dr. K. Sandeep Prabhu, who despite his busy schedule has found time to grace this occasion. We are really enlightened by your knowledge and presence, sir. Thank you, sir.
Thank you. Thanks a lot. Thanks for the word of thanks and kind comments.
Thank you, sir. I extend my thanks to our Honorable President, Dr. B.N. Subbaraya, and Honorary Secretary, Shri N. Chandrasekhar, Shraddha Vilas Educational Institution, Mysuru, for their continued support to conduct this webinar. Thank you, sir. We are grateful to our beloved Principal, Dr. M. Devika, for her valuable contribution, guidance, and encouragements in all our efforts. Thank you, ma'am.
Thank you. Thank you.
My heartfelt thanks to our PG Coordinator and Head Department of Chemistry, Dr. M. K. Manjula, for her kindness and support to conduct this webinar. Thank you, ma'am. I express my sincere thanks to heads, faculties, and students of Shraddha Vilas College, Mysuru, for their valuable contribution in all our efforts. Thank you all. My heartfelt thanks to all the faculties and students of various colleges for their support and presence. I thank you all. Last but not the least, I thank all the participants for your cooperation and presence in making today's occasion a resounding success. Once again, I thank you all for all your kind attention. Thank you all.
Thank you, Rutu. Dr. Sandeep.
Yeah.
Thank you.
Our President, Dr. Subbaraya BM, is here.
Okay. Wonderful.
Yeah.
Very nice.
Very good morning, Professor Sandeep Prabhu.
Thank you.
It's a Yeah, it's a nice to interact with you. At the last summary side, I could join.
Oh,
Pardon me. I couldn't make out anything because I'm a layman with respect to this particular topic is concerned. But anyway, it was a wonderful seminar. And first of all, let me personally thank you for taking your time out of particularly this is a weekend. And during that time, you came on the screen and then helped us and then probably motivated more than 100 participants in and around Mysore, particularly in this area. Professor, I thank on behalf of the Sharda Vilas Educational Institute, and I hope you know that this is 150-year-old institute, a good institute, a prestigious institute in this area. We are doing lots to develop, particularly in the science area, and we have pharmacy college, science college is pretty old, and we have a good alumni network all over the world. We are requesting our alumni to help us and then showcase their work, probably in the area of research or in any area for that matter, to our young students who are willing to work more on these areas. Really, thanks to you for taking a special interest in our college and then agreed to, conduct this webinar. Probably this topic, I heard that it's a very good topic. It motivated many PG students as well as even faculty members also to do their research. I hope that is if the students take out a small leaves out of this and then pursue it in their higher studies or research, I think all of us have done a wonderful work on this.
Yes.
Again,
I totally agree with you. Totally agree with you, Dr. Subraya. Yes. Thanks for your kind comments. You're right. The idea, even though I did not go into the depth, you can peruse through the slides. Dr. Devika has the slides. But what I tried to do was to give a 35,000 feet view of how one can integrate various disciplines within science. You can integrate chemistry, you can integrate biology, you can integrate pharmacology, you can integrate many different things towards a common goal. And I hope I relayed that in different ways today. And I hope that faculty, students take this, because oftentimes when we join hands, we can actually do many things together. And that is what I do here. I lead a team of fantastic colleagues, and each one brings their own expertise. And that complements and then makes us really strong group. And we can do, sometimes it is unimaginable what we can do. And that is because we bring all this expertise that we have and share it and make things, because that is the only way we can actually make science go forward. And in the process, many students, many people, can get really inspired by this and do many things on their own, So I'm really, thankful for this opportunity, because I love to, whenever I come to Mysore, I interact with students in Manas Gangotri, because that's my, alma mater, and I give talks, I interact with students, tell them about, future prospects and so on, and try to help as much as I can, because that's the best thing you can do to give back. But I should tell you, one thing. I'm not an alumni of Sharda Vilas, but my brother is. And yes, and currently he serves as the secretary in the Ministry of Agriculture in Delhi. Yes, he did his POC 1 and 2 in Sharda Vilas. Yeah, so he has very fond memories of Sharda Vilas. Yes.
Wow.
He is the
That's really great.
chairman of the Plant Protection and Crop Improvement Authority in Delhi. He served as the additional director for the PUSA Institute, Indian Agriculture Research Institute. He's a geneticist, plant geneticist by training.
Mm-hmm.
Has developed rice and wheat and other crops and so on. So it's nice because growing up, I've heard so much about Sharda Vilas, and our house is very close to Sharda Vilas. And so every time I'm in Mysore, as I was telling earlier, I see, and and every time I see, there is so much of growth there, so which is nice to see. So I'd be happy to help in any way to interact with your institution, and whatever I can contribute, I'll be happy to to develop anything, or to develop programs, or give my 10 cents of ideas or whatever. I'll be happy to.
Yeah, definitely. Professor, when you come to Mysore, please visit us. You are our special guest. There's no doubt about it. And as you said, a kind of interdisciplinary programs.
Mm-hmm.
I 100% agree with you. That is where the Indian education system is lagging compared to US and Europe. See, for example, in our campus itself, we have pharmacy college, we have science college on one side. On one side, the law college, and then commerce college. I think definitely the faculty members, they can join together to do research, or collaboratively work on a specific topic. Develop the students. That is one area that is we have to ponder around. Now, of course, we have come out with the new education policy. Government of India is working on that, and then the new education policy projects this. They are giving more emphasis on what you call interdisciplinary activities. Just to give an example, a student can acquire a credit by going through a course offered by a US professor. Of course, before that, he has to be recognized, and then certain formality has to be followed. But otherwise, there is a provision to acquire a credit, and then the concerned college has to accept that credit and then go on. That means going forward, it is not a kind of centralized, in the sense it is distributed. Students have more and more opportunities to get acquainted with what's happening around the world. So we are also, particularly within our limited area, we are trying to integrate the pharmacy as well as chemistry, probably the law with the commerce. So this is, we are working on that. And at the same time,
go ahead, please.
I feel that is going forward. This is going to be the future, unless otherwise our students are exposed, in the interdisciplinary activities. There is no future. That is one area. The another area, what we are looking at is, so till now we're looking at the boundaries defined by, could be university, could be the education bodies, probably various education bodies, like in India we have UGC, AICTE, various other bodies. They're imposing certain boundaries. At the same time, even in the universities also, we are trying to think about these. How do we go beyond these boundaries? Because most of our faculty members, or our education governance model itself, trying to work within these boundaries. Sometimes these boundaries are it is really constrained, and then it's put a kind of a constraints on individual minds, so that
Yes.
the exploration of individual topic is lagging behind. Even that is the students are very smart, and then today
Yes.
the information is available at the fingertips,
Everybody.
and they're pretty smart, and then within no time they acquire knowledge.
Right.
But the question is, like in West, we don't have an environment, a platform wherein the students explore themselves. So that is
Mhm.
where I feel that is somehow we have to have a kind of a relationship with certain Western universities and
Right.
Indian universities, where we can create a good win-win platforms, for
Right.
both students
Yes.
as well as faculty members.
Right. Right. Yes. No, I totally agree with you. This interdisciplinary science is what we need, along with basic science, because when you have both, basic science puts, basic, it doesn't have to be science subjects. It can be economics, for example, can be other things, or law, all these puts a very sound, foundation. And then on top of it, now we build these interdisciplinary structures where, for example, you brought up this, right? The law school that you have. The law school could actually think about, for example, science patents, for example, intellectual property dealing with science, patents. That is a huge thing. And if there is somebody who understands the science part of it, but is also a law graduate, I think that is a unique niche area that one could further take on, and that would be very rewarding, I feel. So, and many pharma industries are looking for such individuals, because that's what they need to go through, sift through patents and all these, to see where, if there are any problems and stuff. So that is again a very good example of totally two unrelated fields, but now they are coming together,
Yeah,
and
yeah,
doing something unique. Yeah, and then physics can be, together with biology, and of course we have biophysics, but then it goes even beyond that. Then electrical engineering or electronics engineering can have an impact on biology. And we have some things like that here, biomedical engineering, for example, where bioengineers actually can make certain things that will help biologists like me to carry out further research. And so I think that is how the world is kind of going. And, I'd be happy to talk more about such things, because I really want to see, because, I tell you, since my education has been in India all through from class 1 to PhD, I can tell you that we have a very sound base knowledge, and all we need to do is to just become very creative and innovative, and I think we can do things. Because in terms of math, science, and everything, we can outpace anyone. Any
yeah,
country.
100%. 100%. I agree with you, especially the maths. So Indians are pretty
Oh
strong in
yes.
maths, of course. But now, has maths has taken a back seat, Professor. Earlier, in most of the science colleges, mathematics means,
Yes.
I think professors are pretty strong, and then students also very strong. But now I don't know why the maths has taken a back seat. But of late, of course, India has started many mathematical institutes, must be knowing,
Right.
Chennai
Yes.
Mathematical Institute, CMI, and other.
Right.
And also,
Right.
even in engineering colleges, also they're giving more emphasis on maths. On maths.
Right.
Yeah, that is another area that is we are lagging, because we are not inculcating the maths foundation in all other subject, so which is really required. See, ultimately, whether we like it or not, so we have to work with industries. Industries
Right.
are looking at modeling a particular activity and then how we can,
Right.
experiment at the field.
Right.
So that is how the most industry works. For that, modeling
Right.
itself is really required. For that, there's a pretty strong mathematical foundation is required.
Correct.
That
Statistics
is,
is another. Yeah, statistics. India is actually the world-renowned statistician, Professor C.R.
yes.
Rao. Was a colleague here at our university. I know him personally very well. We used to stay close. Professor C.R. Rao, who did his PhD with Fisher. The Fisher-Rao theorem, comes from C.R. Rao's work. Yeah, amazing. He would have got the Nobel Prize if there was one Nobel Prize for statistics. Unfortunately, Alfred Nobel did not do that. A Nobel Prize in statistics. Otherwise, he would have received a Nobel Prize in statistics. His contributions to statistics are immense. Yeah. So things like statistics require, of course, mathematics, and we can, some of the statistics institutes, ISI in Bangalore, in Calcutta, are world famous.
Yeah. So, that is one. See, what probably my perspective is, a student who came out of various schools, who came out as a graduate, he must have a pretty sound mathematics, and then later on, at the same time, he should understand what's happening in various industries. And at the same time, when it comes to the teaching perspective, I feel that is we need to probably re-look into our system and then see how best we can change ourselves.
Right.
I can give a couple of examples. For example, I was teaching a course at University of South Florida, Tampa campus. About 5 years, I taught a course on computer architecture, of course, nothing. Some of the students from China, some of the students from India, they're pretty good in mathematics. There is no doubt about it. But when it comes to the creating a good business model, where in the colleges we teach a concept and then we try to experiment it by simulation, but ultimately it has to be experimented at the field. For that, we require a good business model. So when it comes to the business model, that is where, of course, we are totally lagging behind. So that is where I feel that is our, the teaching fraternity should be exposed to in various industries of what's happening and things like that. Now that happens, probably we can change the education system.
Correct. Yes, I agree with you 100%. Yes. So I'd be very happy if you would like to things I can contribute towards, please let me know. I'll be happy to.
Yeah, definitely. It's a wonderful interaction with you. I request our principal to get in touch and then at the same time see what best we can closely work on that. We can associate ourselves. You're most welcome. And then at the same time, whenever you visit Mysore, please give it a point to visit our campus.
I will surely do it next time.
Once again, thank you very much. Good day.
Thank you.
Have a nice weekend.
You too. Thank you. Have a good evening.
Thank you.
Thank you. Bye.
Thank you.
Thank you.
Thank you.
Thank you, sir. Thank you, Dr. B.M. Subraya, sir, for the opportunity to listen to such an eminent person. I agree with you as I have personally got inspired. I also thank the principal ma'am for all the support and guidance. I once again thank the speaker of the day, Dr. Sandeep Prabhu, for the informative and inspiring session. Thank you, sir.
Thank you.
Thank you.
Bye-bye.
Thank you, Sandeep.
Thank you all.
Sir,
Thank you all.
Thank you,
very
sir.
much.
Thank you.
Bye.
Sir, there's also a request from participant to share the PDF.
Sure.
Can we share
Yeah,
it, sir?
Yes. Yeah, just make sure
Thank you, sir.
you delete that last picture.
Oh, sure, sir.
Okay. Yeah.
The link for feed. Yes.
Delete those last that last slides, the last picture, and some things that I had after.
Oh, sure, sir.
Yeah.
Thank you, sir.
Thank you.
Box and WhatsApp group, and the recorded session will be available in the YouTube. Thank you, everyone, for joining us today. Have a good day.
Thank you.
Thank you.
Bye-bye.
Thank you. Thank you all.
Thank you. Thank you, sir. Thank you. Thank you, Sandeep. Thank
Thanks,
you.
Devika. Bye-bye.
Yeah. We shall meet again.
Yes, we will.
Thank you. Thank you so very much.
Welcome. My pleasure. It was great.
Thank you.
See you then. Have a good weekend.
Bye. Bye. Vinusha, madam. Vinusha, shall I exit?
Yes, ma'am, you can exit, ma'am.
Okay, I'm taking your leave. If at all there is anything, we'll discuss it later. Okay?
Yes, ma'am. Yes, ma'am. Sure.
Thank you.
Sure.
Thank you. Thank you.
Okay, thank
Thank
you.
you. Thank you.
Thank you, ma'am. Thank you.
Ma'am? Ma'am?